डियर स्टूडेंट्स, आज वाले चैप्टर में हम लोग पढ़ेंगे बायोलॉजिकल क्लासिफिकेशन के बारे में। बायोलॉजिकल क्लासिफिकेशन में सबसे पहले हम पढ़ेंगे कि ये क्लासिफिकेशन कौन कौन से साइंटिस्ट ने किस किस ईयर में दिया। तो सबसे पहले हमारे पास आएगा, दिस इज थ्री किंगडम सिस्टम विच वाज गिवन बाय Haeckel. Then we have this five kingdom system that was given by Whittaker in 1959. Then Carl Woese, his name was Carl Woese. He gave six kingdom system. Then again Carl Woese gave three domains for all these kingdoms. There was one more scientist, that is Linnaeus, who gave two kingdom system. So first of all you should retain the names of these scientists. Likewise, first of all, you can write down this: Haeckel 1894, Whittaker 1959, Carl Woese Karl, Carl Woese 1977, again Carl Woese 1990. So this was about the basic pattern of classification, scientist name and their year. Now second part, we have this simple this tree, the classification tree. We have prokaryotes, bacteria and archaebacteria. Then we have these four remaining eukaryotes, that is animalia, fungi, protista and plantae. After that, we are supposed to know the difference between these three, that is in eukaryote, eubacteria and archaebacteria. Here we have this eubacteria and archaebacteria. They are associated with they are basically they are monerans, and they are prokaryote, and this is a eukaryote. So eukaryote, as I told you, eukaryote has four kingdoms, and here prokaryote has these two. So these are the simple differences, 70 years, 80 years. So you have done these already. Then again, an important part is the difference between the gram negative and the gram positive. So generally question comes from with respect to their cell wall. So their thickness, their periplasmic, peri is around, then their lipid contents, teichoic acid and peptidoglycan concentration. So you can have a glance over this, that what is absent and what is present, what is in small amount and what is in large amount. The next part is the very important part, that is human diseases caused by bacteria. Why I am telling it very important? Because same type of content is a part of syllabus in human health and diseases, that is part of +2. So you are supposed to retain, you are supposed to learn, you are supposed to cram all these diseases and their causative agents. For example, we have cholera, Vibrio cholerae, typhoid, salmonella typhi, tuberculosis, mycobacterium tuberculosis, leprosy, mycobacterium leprae, pneumonia, diplococcus pneumoniae, plague, Yersinia pestis, diphtheria, corynebacteria, diphtheria, tetanus, clostridium tetani, food poisoning, that is again botulism बोलते हैं इसको, that is clostridium botulinum, and syphilis, that is treponema pallidum. So you can have a glance over this. These are again very important, that human diseases, कौन कौन सी human diseases है, कौन कौन से causative agents हैं, ये बहुत बार पूछा जाता है, कई बार डिफरेंसी में पूछा जाता है कि which of the disease is caused by bacteria, which of the disease is exclusively caused by viruses. So in that case also you can find out the solution of those questions. So you can have you can learn them. Then we have similarly we have a plant disease caused by bacteria. So this portion is also a part of syllabus in case of applied biology, that is the rice, apple, carrot, citrus, cotton, potato and potato. कौन कौन सा नेम ऑफ द डिजीज क्या है, बैक्टीरियल ब्लाइट्स है, बिकॉज ब्लाइट इज आल्सो कॉज्ड बाय fungi. So you can compare these xanthomonas, arvenia, then clavibacter and streptomyces, scabies for scab in potato. You can note down if you want to note some part. Then coming to the further, very important, economic importance of bacteria, because it is again a part of applied biology in plus 2, that is ammonification. Ammonification, which are the bacteria which are responsible for ammonification, we can know their names, and what is the what is the role of these bacteria, or what is the definition of ammonification. So here you are supposed to retain them, that what is, only read them. Second is nitrification, that is caused by two very important. This is the most important. One is the nitrobacter, and other is nitrosomonas. So nitrobacter क्या करता है? It converts ammonia into nitrites, and nitrosomonas क्या करता है? It converts nitrites into nitrates. So you are supposed to retain them. They are again very important for competition. Then fixation, nitrogen fixation. Nitrogen fixation can be azotobacter, clostridium, and rhizobium. So you are knowing the details of rhizobium. That's again an important part in mineral nutrition chapter, part of plant physiology in plus one. And what is the role of their converting atmospheric nitrogen into organic nitrogen? Nitrogenous compounds are also oxidized to nitrogen. All these activities of bacteria increase soil fertility. So it is economic importance. Moreover, economic importance, as I've told you earlier, economic importance does not mean only which is positive for the economy, which may be positive or negative. That affects the economy. Coming to next, antibiotics, again a very important. What is antibiotics? क्या क्या antibiotics है? तो बहुत डिटेल में आपको जाने की जरूरत नहीं है. You are supposed to know only the name of the antibiotic and their source. Antibiotic, their source. Antibiotic, their source. Antibiotic, their source. Antibiotic, their source. You can note down this. First is streptomycin, then is aromycin, chloromycetin, bacitracin, and polymyxin. Uh, Now coming to the next part, that is the industrial uses. Again a very important part, because it's also part of microbes in human welfare. That is plus 2 part. That is lactic acid, butter, cheese, curd, yogurt, vinegar. So their role of the organisms which are used, that I've told you, that is microbes in human welfare. And what is their role? How they are formed? So you should at least note down the names of these products with the name of these bacteria. Now coming to the next, again these are the products, that is alcohol, rating of fiber, very important. This question has been come many times in a competition. Rating of fiber, that is clostridium tertium. The fibers from the fiber yielding plants are separated by action of clostridium, is called rating of fiber. Then again it's a very important, curing of tea and tobacco, that is Mycococcus candescens, Bacillus megatherium. So you can note down these two. Now, animal disease caused by bacteria, sometimes it's also because it's also part of animal husbandry also, that is sheep. We have anthrax, cattle, brucellosis, that is disease का नाम cattle, bovine term is used for this cattle, that is cattle, black leg. Then the name of the pathogens: Bacillus anthracis, Brucella abortus, Mycobacterium bovis for bovine, and Clostridium chainvii. So note down this. Coming to the next part, that is the difference between bacteria and cyanobacteria. So this question has been asked so many times regarding their differences. So keep one thing in mind, that you consider that cyanobacteria is near to plant, that is they are like plants, so most of the parts are relatively like plants. So plants with they are oxygenic photosynthesis, and they are anoxygenic photosynthesis. This is important. Second is they are like animals, that glycogen is the reserve food material, whereas here the reserve food material is starch, that is known as cyano is blue. They are always aerobic. They can be aerobic or anaerobic. They are autotrophic. They can be aerobic or heterobic. They may possess flagella. They lack flagella. ये छोटे छोटे साइज में होते हैं, ये बड़े साइज में होते हैं. This is the most important. So note down all these contents parts, the differences. And another name of cyanobacteria is also known as blue green algae, as I've told you earlier. So that's all.